All right, you guys, hello and welcome back. Sorry if I'm not as movement inclined with my arms today. You can see I got a little fluffy butt in my lap. So he wanted some attention. I just couldn't say no to his nose face. So we are going to continue and actually finish off chapter 13 today. It's a pretty short chapter. There's a lot in it, but as this is typically our last chapter of the year, we generally aren't going to go into a whole lot of detail on it. We kind of save that for some of the other stuff. For computers. I know, the light. So this is going to look at sections, that's actually a typo, sections 3 through 5. I promise I will fix that in the Prezi. My word to it. So sections 3 through 5 notes, and then we have our additional resources at the end. So without further ado, I'm going to move my camera back up there. And without further ado, let's get going. Let's get crack-a-lacking. All right, so section 3 is on atypical number of chromosomes. Now, atypical means not typical. Now, normally, humans, our chromosomes come in pairs. We have 46 total chromosomes or 23 pairs. And when a cell has that normal chromosome number, we call it a euploid cell. Just means that everything is A-okay. If there are more or less chromosomes than 46, so if you have 45 or 47, it can lead to either spontaneous abortion or it can lead to different disorders and syndromes. And we'll talk about some of those in just a moment. So euploid is when you have the correct number. And if you look off to the side, we have polyploidy and aneuploidy. So we're going to discuss those, how they relate, how they're different. So let's get going. So polyploidy is when a cell has extra chromosome sets. So it's not just one or two extra chromosomes, it's every single pair has extra. So what this could result from is fertilization by an oocyte, either by two sperms, so two sperms get in there at the same time, or if a gamete was left diploid, so it never went down or never finished meiosis and went down to a haploid number. Now this is very, very severe in humans. Humans cannot survive this. So if this does happen, whether it's two sperm fertilizing one egg or having a diploid gamete that is fertilized or does the fertilizing, it doesn't matter. Our cells don't know how to read that and create a functional human being out of it. So this actually is a common cause for spontaneous abortion. Now that's essentially a miscarriage. It's just when the body rejects it because there's something wrong with the zygote. So for us, for humans, our euploid number is 46, 23 pairs. So we have diploid chromosomes. Now haploid, that's what our gametes should be, but there can also be triploidy or tetraploidy. So what that means is in triploidy, instead of having pairs, you have triplets of each chromosome, or tetraploid, you have 4 of each chromosome. Now again, that's not gonna be something we see in humans. Although there are some plants like strawberries that are polyploidy. They have multiple of each chromosome. I can't remember, I think it's somewhere between 4 and 7 of each chromosome. All right, so aneuploidy. Now this is when a cell will lack a chromosome, not sets, just one individual chromosome, or it can have an extra chromosome or 2. So you're going to be either missing 1 or 2 chromosomes, or you're going to have 1 or 2 extra chromosomes. And this comes from something called non-disjunction. It occurs during meiosis. And what this means is instead of the chromosomes or the sister chromatids being properly separated during meiosis 1 and meiosis 2, you end up with one chromosome that has 2 copies of it. So let's say, I need this hand rudely. You have 2 copies of chromosome 21 in this cell, and this is our egg. You have 1 copy of chromosome 21 in this cell, which is the sperm. They come together, they fertilize, we now have 3 copies of chromosome 21. Individuals with trisomies are more likely to survive those than those with monosomies. So trisomies is when you have 3 of a given chromosome. So like we just mentioned, trisomy 21, you have 3 of the 21st chromosome. Generally speaking, unless it's a sex chromosome, you really can't survive with just 1 of any chromosome. That would definitely lead to a spontaneous abortion. And certain chromosomes, especially the larger ones in like the first 10, if you have trisomy of those oftentimes will also lead to a spontaneous abortion. Sex chromosome aneuploidy, it tends to be less severe than autosomal. A lot of that just has to do with how the sex chromosomes themselves work. Specifically how males only have 1 X chromosome anyways, and females, we have X inactivation. So we do tend to see it's not as severe when it's the sex chromosomes being affected. All right, some examples. The most common autosomal aneuploids are trisomy 13, 18, and 21. So that means that you can have 3 copies of the 13th chromosome, 3 copies of the 18th. Those do tend to be more severe than trisomy 21. Now, what trisomy 21 is, that's Down syndrome. So 3 copies of the 21st chromosome tend to lead to Down syndrome. Sex chromosome aneuploids can include XO, meaning that you have an X chromosome, and that's it. No second X, no Y. So that's a monosomy. That can lead to Turner syndrome. XXY can lead to something called Klinefelter syndrome. Triplo X females, which are XXX, and XYY males, which are Jacobs syndrome. Now the picture at the bottom just kind of talks about like what's going on with non-disjunction. So we have 46 chromosomes. We go through DNA replication. It says 92, but really it's 92 sister chromatids at that point. We don't think of the chromatids as individual chromosomes while they're connected. Then we go through, we end up not separating properly. So we should have, or we start with 1 blue, 1 green, we duplicated, separate, one of the blues does not move over here, so now we have a cell that's missing that's chromosome and a cell with an extra chromosome. That's the basics of what's going on. So I'm in that awkward phase of day when like light is just right. All right, so section 4. We talked about atypical chromosome number. Let's talk about some atypical chromosome structure. So chromosomes have very, very specific shapes. Their genes are located at very, very specific locations in their or within their shape. So their shape and structure is very, very important. And any change to that can have massive, massive effects. Whether it's a deletion, which is exactly what it sounds like, you lose a piece of information. That's not digging into you now. How's that, Ripley? There you go. All right. See? Now you're out of the light. Sorry. So, deletion is when you have a portion that's actually cut out and removed, like this picture at the bottom here. So this is what the chromosome should look like, but part of it is missing, so it's now shorter than it should be, and you're missing that entire section, which could easily be multiple genes. Duplications can also occur. That's where you get an extra copy of a section, not necessarily of a gene, of a section. And again, there can be multiple sections or genes within that section. We see here, this is a standard length, and we still have it there, but then we have another copy of it there, causing the chromosome to be longer than it should. These can occur when there are translocations or inversions, and these will definitely disrupt the pairing of chromosomes in meiosis, which means that they may not separate properly, and it can just easily be a hot mess. Now, if you're wondering what an inversion is, take the section out, flip it around. So if I was to take my torso out, flip it around, then my butt would be right here underneath my head, and my sternum, my neck, my chest bone and all that would be down lower. Not a pretty picture. It's in the right location, but it's not in the right orientation. All right, let's talk a little bit about translocation. Now, there's actually different types of translocations. We're only going to talk about a couple of them. So the first one we're going to talk about is called a Robertsonian translocation. So this is a piece of one chromosome breaks off and attaches to another chromosome. So imagine if I am a chromosome. My husband is a different chromosome. We are not homologous. His arm breaks off and attaches right here on my arm. So I have all of my information plus this extra information from him. So I gain extra stuff, extra information. So I get some new information. He is missing some information. Let's see. And then we have reciprocal translocation. This is where you have, again, non-homologous chromosomes, and they exchange parts. So it's like crossing over in meiosis, except it's not between the homologous pair. So again, if my husband and I are non-homologous chromosomes, I take my right arm off, I give it to him, he gives me his left leg. We both exchange information. Neither one of us has chunks missing, but we have incorrect information left on us. So translocation that deletes, like we said with the Robertsonian, how it can actually just be taken out and not replaced with anything. Duplicates is when you can have a Robertsonian translocation even between homologous, but there's not the equal exchange. Everything just goes one direction. So it can delete, duplicate, or disrupt genes. And anything that's going to do that is going to definitely harm your health. Again, there's any number of disorders that can occur. Notice I'm not giving a whole lot of examples because it all depends on exactly which chromosome, where in the chromosome, how much of a section is moved. There are so many variables, it's really hard to give specific examples for each one of these. Translocation carriers may have a normal phenotype, but they may have affected children. So what that means is a person who maybe has one completely normal chromosome and then one, let's say for chromosome number 16, one version of their chromosome is completely normal. There you go. And then the other one maybe has a small extra part added to it from leftover from a parent. So they have this small translocation, but they're considered a carrier for it, and they don't express it. They are not expressing any health issues from it whatsoever. However, they can pass it on to their child, and their child can now have certain health issues, disorders, syndromes, etc. Again, it all depends on where the translocation is occurring, what type of translocation, lots and lots and lots of variables. Inversion is caused when a segment of a chromosome actually flips around. So if you look at this, we have A, B, C, D, E, then we have our centromere, F, G, H, I, J. What happens is this section here, G, H, I, flips around. So A, B, C, D, E, F, we got our centromere, F, I, H, G, J. So that may cause some health issues. Again, it all depends on exactly where in the chromosome it occurs, as well as how big of a section it is. Because we have a lot of those sections, such as signs and lines, where if it occurs there and just flips around, it's just repeating sequences anyways. And there's also palindromic sequences mixed in there. It may not have any effect whatsoever, or it might have a very minute effect. But if it's in an area that codes for something pretty darn important, like different neurological functions, then it actually could have larger health effects. Yes, hi, Ripley. We also have something called isochromosomes. So what an isochromosome is, it's where you have your two sister chromatids, and instead, so here's a chromosome and chromatid. So we know that during meiosis, they should separate like that. So I have the hand and the arm. But what happens in an isochromosome is that it doesn't split along the normal plane. Instead, it splits perpendicular to it. So instead of having hand, arm, I have two hand section and two arm section. So it's really hard for me to explain these. I'm not sure why. I've been pretty tired today, so I do apologize. But look at the picture. So our original chromosome, the short arm is green and the long arm is purple. Goes through DNA replication, it replicates, and now we have our two sister chromatids. Now what we would expect during meiosis is, specifically meiosis 2, when we split those sister chromatids, they should each look like this, green on top, purple at the bottom. But instead of splitting up and down, it splits left to right, horizontally. So we get a chromosome with two lower arms, or two long arms, no short arms, and a chromosome with two short arms and no long arms. So we're missing huge amounts of information with this. And we have duplicated sections, quite a few of them actually. And not only that, but they're both going to be, each side of that, it's going to be inverse from the other, because all we're doing is we're taking these and moving it to the top. So it's going to be in, the two sides are essentially mirror images of each other. So there's going to be some inversions, and we're missing entire sections of chromosomes at this point, which could once again be major amounts of genes. Ring chromosomes. So ring chromosomes are exactly what they sound like, chromosomes that form rings. So ring chromosomes arise when telomeres are lost, and they leave these things called sticky ends. Now, what these sticky ends are, they're the broken area where it wants to, so the broken area of a chromosome, and it wants to rebound, it wants to fix that broken area, like you can see here. So here's the full chromosome, and in the course of losing some of the telomeres are already gone, and the DNA still tried to replicate, and the cell still tried to divide, and we end up losing this section and that section. Well, these sticky ends, see how they're all jagged? They don't want to stay like that. They want a nice complete end. So what they can actually do is fold over on itself and close up. This can form a ring then. Sorry about that. And then we have two little broken off sections that just kind of reform to make their own thing. So ring chromosomes can produce symptoms when they result in additional genetic material, because again, you have duplications of different things or of different genes. It can have massive effects depending on exactly which genes are being affected and to what degree it is forming that ring. All right, section 5, uniparental disomy. All right, so uniparental disomy, or UPD, is what happens when two chromosome or chromosome parts are inherited from the same parents. So what that means is when we have our 46 chromosomes, 23 pairs, we expect one of each pair to come from each parent. So one should come from mom, one should come from dad for every chromosome pair. But maybe there's a chromosome where you have 2 copies of that chromosome from mom, but no copies from dad. So it doubles one parent's contribution for that particular chromosome. And pretty much how this could arise is from double non-disjunction. So each parent went through non-disjunction. So let's say we have, remember we said that during meiosis, you want to separate your sister chromatids, but what if you have, let's say, an egg where with 2 copies of a chromosome, chromosome 19. Now, if it was to be fertilized by a sperm that went through meiosis correctly, it would lead to 3 copies of chromosome 19. But what if it was fertilized by an by a sperm that also went through non-disjunction, but this time it didn't have any of it? Remember, when we split it, we split chromosome 19 during meiosis, you end up with 2 gametes, or in this case, I'm showing you 2 gametes, each with 1 copy of it. But if it doesn't split properly, that means you have a gamete with twice as much and one with nothing. So we're saying that a sperm that also went through non-disjunction, but this time didn't get any copies of chromosome 19, fertilizes an egg that has 2 copies of it, and now you have all of chromosome 19 from mom with no contributions from dad. So, remember if I had that explained down here. So UPD can arise from a trisomy and subsequent chromosome loss, or from 2 non-disjunction events. So that means the 2 non-disjunction is what I already gave you the example of. So the egg had non-disjunction and ends up with 2 copies of a particular chromosome. The sperm went through non-disjunction and ended up with no copies. They fertilized. They still get 2 copies, but they both come from the same place. Another example could be if there is non-disjunction on just one part. So the egg maybe had non-disjunction, has 2 copies of chromosome 19. We have a normal sperm come along, fertilize. We have now 3 copies of chromosome 19, but for whatever reason, one of them gets lost. And it just so happens to be the one from the sperm. Now you still have 2 copies, both from mom. So UPD can cause disease if it creates a homozygous recessive condition associated with an illness. So by itself, the process or the results of UPD don't necessarily mean it's going to be bad. It just means you have 2 identical contributions. However, if there is an allele, a recessive allele for cystic fibrosis, and mom didn't have cystic fibrosis, she was a carrier, but there's a bug buzzing around up front, up there, sorry. But she, as being a carrier, she does have the recessive allele, and she goes through non-disjunction with her eggs, and 2 copies of that recessive allele end up in her egg. Well, if it ends up being UPD and the contributions only come from mom, then that means that child can now have cystic fibrosis because you got 2 recessive alleles from mom. Let's see, UPD can also cause disease by disrupting genomic imprinting. That's something we talked about in a previous chapter, how the exact same mutation or allele this exact same mutation can cause different effects depending on which parent passes it on. So we gave, in a previous chapter, we gave the example of Engelmann syndrome and Prader-Willi syndrome. They're caused by the exact same deletion on the exact same chromosome, but it ends up with 2 very different and distinct disorders depending on if you get it from mom or dad. So because this limits your contribution from either mom or dad, it can thus affect the genomic imprinting. All right, and last but not least, additional resources. If you have any questions about this or kind of want to go into a little bit more, I would definitely suggest that. Yeah, let me get out of the sunlight. No, not gonna work. There we go. Oh yeah, now the clouds come. All right, so the first video, it starts off mostly talking about pedigrees, but there is some information on karyotypes at the end. So definitely maybe go ahead, check that out if you have more questions about karyotypes. Yeah. There it is. All right. This one will also be a good one to check out. We talk about chromosomes. We talk about structure and all that good stuff. I would definitely recommend checking this one out. It'll be good for you too. And also chromosomal mutations. This would be an excellent one to check out, specifically because I do also talk about like translocations and all that. So if you weren't quite sure what was going on with that and you're too scared to ask me because you're being silly, check this one out. It's a good resource. I'm not just saying that because I made it. We also have a whole bunch of other videos. I'm not going to go through and kind of click on each one. I'll just tell you what they are because there's 6 of them. Amoeba Sisters has a great one on chromosomes and karyotypes. So again, they're silly, they're cartoony, but they break things down very well. A group called Clearly Stated with their video called What is a Chromosome? Biology Tube has one on chromosomal mutations. AK Lectures has one that specifies, I don't know if that's the right term, but we're going to go for it. Specializes, there we go, in chromosomal deletions, inversions, duplications, and translocations. So there's a lot with that, not just the trisomy, monosomy chromosomal mutations, but more the trickier ones. Photobiology, chromosomal abnormalities, aneuploidy, and non-disjunction. So if you're not quite sure what non-disjunction is, definitely check that one out. And then National Cancer Institute has one on understanding uniparental disomy. So I definitely recommend checking those out. They'll be good for you. They'll have some good information. And let's see. Otherwise, if you have questions, hit me up in our Google Classroom or send me an email. You know how to get in touch with me. I will also post this link up when I post or in the description of the videos so you can have access to the additional resources. So the Prezi link will be posted. If you do have any questions, please let me know. I'm not psychic. I just play one on TV. But in the meantime, I hope everything is going well. Everyone's staying healthy. And in the meantime, you guys, take care of yourselves, all right? All right. Bye-bye.